Now, so far, your recombinant DNA is prepared. Your recombinant DNA is introduced into the suitable host. And now it is dividing, the suitable host is dividing, and likewise your plasmid DNA is also dividing. Its multiplication is taking place. One multiplication is that when the cell divides, DNA also divides. Another multiplication is that within a single cell, there are multiple copies of these plasmids synthesized and produced. Okay, it's division. You can say this is DNA replication taking place inside the cell, right?
Yes.
Now is how, I mean, the question that I left you with was that how do you determine that it has to be the second segment? When you are chopping this DNA off, you get 3 segments, and your gene of interest, GOI, gene of interest, is the second fragment. So how you are going to pinpoint or dictate this vector DNA to only ligate with the second fragment and not with the first and third fragment? Is there any strategy that you can come up with? Jairus, turn on your camera and let me know.
Yes.
Is there any way
Uh,
that we can, direct this vector to only pick up this one and not these two? There's no such way.
uh,
There's no
no.
such way. See, these are very small molecules.
Cutting,
Yeah.
cutting by
Cutting to is there.
okay.
Cutting only generates 3 fragments. Like, you cut it at 2 places, you'll get 3 fragments. But how are you going to pick a specific strand? See, for this, we have separation and isolation of DNA fragments. There is a technique. This is molecular biology technique. You call it as molecular technique. And a very interesting one. What you can do is you cut the DNA by restriction endonuclease enzymes. You get the fragments. And now these fragments should be separated by the technique called gel electrophoresis. Gel electrophoresis is a technique to separate and isolate the DNA fragment. Okay, now what happens? There are a few basics again to understand. The DNA is negatively charged. DNA has phosphate group, and phosphate has the negative group, negative charges on it. So your DNA molecule, no matter how big, how small, circular or linear, doesn't matter. All the DNA will have negative charges on it, right? And then when you want to run it, there are a few components of this technique. First is this gel. It is the agarose gel. Agarose gel is produced from, it's actually a natural hydrocolloid. The term we use is hydrocolloid. That is a substance that can hold water, water holding substance. Okay, hydrocolloids are water holding substances. These are obtained from red seaweed. What is red seaweed? It is actually the red algae. Red algae. Okay, red algae, it's cell wall. Outside the cell wall, you have a layer of agar, algin actually, from which agarose is derived. It's a heteropolysaccharide. Agarose is a heteropolysaccharide, and it is available as white powder. So when you mix this white powder with water or in a buffer solution, it will not get dissolved. It will not form this jelly-like substance. You'll have to warm the water. You heat it up to 60 degrees Celsius, and then the powder will completely melt away and will get dissolved in water. And then when you allow it to cool down, it will solidify like a gel. This is how you use, gelatin and agar at home also. Right, we use it for jellies and preparation of ice creams at home even. So these are what? These are hydrocolloids. They can hold water, and they can form jelly like substance. So agarose gel is also the same thing, but this is for the household usage. You do not have a lot much purified components. Here it is very, very purified, like it's 99.99% pure agarose, so it's very expensive. Right, now how do you prepare the gel? See, you have dissolved it. Okay, why did I erase it? It's agarose gel. Now, when you prepare this agarose gel, you have choice to go for 1% agarose, you go for 2% agarose, or 2% will be a lot. Let's keep 1.5% agarose, or you go for 0.5% agarose. You have choices to go for any percentage. What does that mean? For example, if it is 1%, you have 1 gram agarose powder in 100 ml water, buffer. It's 1.5%, it's 1.5 gram in 100 ml, 0.5 gram in 100 ml. Okay, these are the different composition. Now, what is the meaning of percentage of agar? This means that you have control over the percentage of agarose powder that you are going to dissolve in water. But why? What is this percentage doing? More agarose means, like, I'm going to draw 3 diagrams for you. You identify which one is 0.5, which one is 1, and which one is 1.5. Which one is 0.5?
The first one.
The first one. So you identified, you understood it. This is 0.5, this is 1%, and this is 1.5%. So more agarose means that agarose actually makes a network-like thing. Okay, it creates some pores. The molecules are arranged in such a way that you'll have certain pores created. Now, from outside, you won't be able to see the pore. The pore is big enough only for the DNA to pass through. It's not big enough for you to see, right? Now, if let's say, now imagine that you have DNA molecule. DNA molecule to pass through these pores, will it be easier, or its passage through these pores will be easier? Of course, it is going to be more easy for 0.5%, right? Yes or no? Because it has got large pores. It has got large pores. It has less hindrance. It's like, the traffic. You are going to school and you have a lot of traffic on road. You cannot, your speed is limited. Your speed is reduced because you cannot, just pass through these many cars and vehicles and automobiles. Likewise, DNA also cannot pass through these many agarose molecules. So therefore, its speed is retarded. It's slowed down. But there's one more factor that governs it. Larger is the DNA fragment. Larger is the DNA fragment. Slower is its movement. Smaller is the DNA fragment. Faster it moves in on the agarose gel. Okay, so speed and length, it has a reciprocal relationship with each other. We can say that, we can write here, smaller DNA is faster and larger DNA is slower. So now when, size-wise also you understood. Now, if you have DNA fragments of very, very small sizes, to resolve them, you'll use 1.5%. Now, imagine that this is, a 1.5% gel. I'll be deleting it for each. I'm just trying to explain you. Let's say that your fragment size is all very small. You have 100 base pair fragments, which are very, very small. So you have one is 100, another is 150. The difference is just 50. So when you run the gel, they'll be very close to each other, very, very close to each other. And you won't be able to, differentiate them. In that case, you'll have to increase the gel percentage so that their movement is retarded, but they move slowly and they also get separated over the time. There is resolution between the two fragments, okay? But if you have large fragments, you don't have to go for 1.5% gel. You can simply go for 1% gel. Ideal choice always, in case of DNA gel electrophoresis, an ideal choice is of 1%. Generally, we go for 1% DNA. What happened? Yeah. Generally, this is our preferred agarose strength. But that's not all. That's not all. So when you cast the gel, let's say this is known as gel casting, you prepare the solution, you warm it up at 60 degrees, you melt the agarose down. And now on a casting tray, you will seal this end, you will seal the opposite end also. And here you'll put a comb that is literally like a comb. Okay, it's just like this. Why you have a comb, placed here? You'll place this comb here at this position. Why? To create gels, to create wells. This is known as a well. Okay, where would you put your DNA unless you have wells? You see, wells are also shown. I have another image also showing wells. This is not very clear. Okay, I don't have any other. You check this here. These are wells. The faint wells are shown. These are wells. These are the wells where you will actually put your DNA. Correct? Now we'll say that your DNA, let's combine all these information so far that we have got. Your DNA is negatively charged. You have agarose percentage. Depending upon the length of DNA strand, you choose the correct percentage for your agarose. And you know that the negatively charged DNA, if it is smaller, it will be moving faster, and larger DNA will be slower in movement. So if you have a mixture of DNA fragments, one fragment is 100 base pair, another fragment is 1000 base pair. Naturally, after running the gel, your 1000 base pair will be upper, and it will be occupying the uppermost region than the 100 base pair. At the end of the gel, you will have 100 base pair. At the top of the gel, you will find the 1000 base pair.